Now, before we get started, we need to talk a little bit about microscopy and how it has led to some false assumptions and views of cellular function and how organelles may be working. Okay, say this bagel is an organelle in the cell. As you can see, it's round, it's three-dimensional object with a hole through the center. However, when we look at images of a cell under the microscope, we're typically looking at a two-dimensional image. That is, we're looking at sections through the cell. Okay, some modern microscopy techniques allow us to view cells in 3D, but all the early techniques were two-dimensional. So, let's think about this bagel and what happens when we section it. If we section it through here, and then we look at the two sections we get, they will look like this. Spaced equally apart, but they look like two small circles. So we could conclude that the bagel is two small circles. However, if we section our bagel across here, then when we look at it down the microscope, what we would see is a shape like this. So we would conclude that the bagel is in fact oval shape. Or alternatively, if we slice the bagel through here, then when we look down the microscope, we may see something like this. However, from this image, we don't get the full picture. We don't see what's behind it. We don't know if it's a long hollow tube extending out behind or if in fact it stops abruptly. And that's one of the problems with early microscopy. We just don't know what the shape is, and some false conclusions have been derived from looking down the microscope. As you can see, there is a problem. The simple bagel, depending upon how we section it, can be many shapes and structures. And the same has happened in cell biology as a result of sectioning cells and tissues. We have some odd ideas of what the structures look like in the cell, and this in turn has led to some false assumptions about function. But there's another problem, and that is that the cells are not living but are dead when viewed. Hence, effectively, you are viewing a snapshot of the cell and not how it functions. For example, suppose you set up 3 cameras in a field. Cameras 1 and 2 diagonally opposite each other in the corners, and a third camera in the middle of the field. And say that field had a horse in it. Now, suppose the horse was standing in front of camera 1 and triggered it to take a photograph. The horse would have all 4 feet on the ground. Say the horse then runs across the field and triggers the camera in the middle of the field, and then stops at the far side of the field where it triggers the final camera. You would now have 3 photographs of the horse. One on one side of the field with all 4 feet on the ground, one in the middle of the field where the horse would be running and could have all 4 feet off the ground, and a third on the far side of the field where the horse again has all 4 feet firmly on the ground. From these photographs, you could conclude that the way horses move is that they stand on all 4 feet, lift all 4 feet into the air, fly and hover across the field, and then when they get to the far side, lower their feet so they can stand again. And this is the danger of just looking at snapshots of live events happening in the cell. You don't get the full story. So what I'm trying to say here is that when you're looking at images taken down a microscope of a cell, remember the cell is dead and has been fixed with chemicals such as paraformaldehyde or glutaraldehyde or equivalent that cross-link the proteins. Therefore, you're just seeing a snapshot in time of what was happening in the cell. Plus, you've got the issues of the cell being sectioned, so you don't see the full 3D image. And unfortunately, this has led to some misconceptions of how organelles work in the cell and their structures.